Welcome students. So our topic for today is translation. The slide defines translation. Let us look at the first point. It says translation is the process of decoding the mRNA into a polypeptide chain. This means after mRNA is made from DNA, the next job is to read its coded message and use that information to build a chain of amino acids, which is a polypeptide. The next point talks about the machinery. Ribosomes read mRNA 3 bases or 1 codon at a time and construct the proteins. So, the ribosome is like a factory. It moves along the mRNA strand, reading the genetic instructions in sets of 3 nucleotides called a codon. Each codon specifies one amino acid. And finally, the last point summarizes the core idea. Translation involves converting the nucleic acid language to protein language. Essentially, it is the process of changing the code written in the RNA alphabet, AUGC, into the functional language of proteins, which is made of amino acids. Moving on to the next slide, which continues on translation. The first point here says the decoding of mRNA is carried out by a submicroscopic machine called a ribosome. So, this ribosome is a tiny molecular machine that binds to the mRNA and starts the translation process. The next point explains its action. The ribosome moves along the mRNA, reading the message and synthesizing a new polypeptide chain. Imagine the ribosome sliding along the mRNA strand like a reader moving along a sentence, and as it reads each word, which is a codon, it adds the corresponding amino acid to the growing chain. Now, the slide also distinguishes between two processes. Transcription occurs when DNA acts as a template for mRNA synthesis. That's the first step, making the mRNA copy. And then, translation occurs when the sequence of the mRNA codons determines the sequence of amino acids in a protein. So, transcription is about copying the code, and translation is about using that copy to build the protein. Now, looking at slide 4. This appears to be a diagram or a flowchart. It shows the title translation and then indicates movements from the nucleus into the cytoplasm for synthesis. This diagram is helping us visualize the location. It tells us that the process of translation does not happen inside the nucleus where DNA is. Instead, the mRNA travels out from the nucleus into the cytoplasm, and it is in the cytoplasm where the ribosomes are located, and that is where protein synthesis or translation actually takes place. This slide has a very helpful schematic. Let us understand it step by step. At the top, we see the flow. Double helix DNA leads to transcription mRNA, which leads to translation polypeptide. This shows the central dogma, from DNA to RNA to protein. Below that, we see 3 codons listed: codon 1, codon 2, codon 3. And they are linked to the peptide chain structure NCC-NCC-NC. Each C connects to an R group, and the labels show us an example: arginine, tyrosine, tryptophan. So, this diagram is beautifully explaining that each set of 3 bases on the mRNA, like a 3-letter code, corresponds to one specific amino acid. These amino acids are then linked together in a chain by peptide bonds forming the beginning of a protein. Now, slide 6 introduces us to accessory proteins. The point says they are called factors. These are helper proteins that are essential for translation to happen smoothly. They are required for the 3 stages of translation, which are initiation, elongation, and termination. For initiation, we have eIF, which stands for eukaryotic initiation factors. For elongation, we have eEF, the elongation factors. And for termination, we have eRF, the releasing factors. So these factors are like specialized assistants, each helping at a specific stage of the protein building process. Now, slide 8 is titled Step 1, Initiation. This is a simple diagram showing the first step. We can see the mRNA transcript. The start codon AUG attaches to the small ribosomal subunit. Then this small subunit attaches to the large ribosomal subunit. So initiation is all about assembly. The mRNA, the small ribosome piece, and the large ribosome piece all come together at the correct starting point on the mRNA, which is the AUG codon. The next slide gives more detail on step 1, initiation. It tells us that this phase requires over 10 eukaryotic initiation factors. These factors have specific jobs. First, they are required to recognize the cap at the 5' end of the mRNA. This cap is like a special marker that identifies the front end of the message. Then they help in binding the initiator, which is the methionine tRNA, to the small 40S ribosomal subunit. Finally, this whole complex scans along the mRNA from the 5' cap, moving downstream until it finds the first AUG sequence. This AUG is the start codon, the green light to begin the protein. Continuing with initiation, slide 9 explains the final setup. The start codon and the initiator tRNA must be correctly positioned in the P site of the ribosome. The P site is like the first docking station. Once the mRNA and this initiator tRNA are correctly bound, the large 60S subunit joins to form the complete 80S initiation complex. This joining causes the release of the initiation factors, or eIFs. Their job is done. Now, with everything in place, the ribosome is ready, and the next stage, which is elongation, can proceed. Now, we move to step 2, which is elongation. This slide has a diagram. We can see the ribosome on the mRNA. Two tRNAs with the amino acids are in the A and P sites of the ribosome. A peptide bond is formed, joining the amino acids. We see a growing polypeptide chain. So, elongation is the repetitive cyclic process where the protein chain is actually assembled one amino acid at a time. Slide 11 elaborates on the elongation process with key steps. The first step is the transfer of the correct amino acyl tRNA from the cytoplasm to the A site of the ribosome. Next, a peptide bond is formed. An enzyme called peptidyl transferase, which is actually part of the ribosome's rRNA, forms this bond between the amino acid in the P site and the new amino acid in the A site. This lengthens the peptide chain. Then, translocation happens. The entire ribosome shifts. The new peptidyl tRNA, along with its mRNA codon, moves from the A site into the now free P site. This movement frees up the A site for the next incoming amino acyl tRNA. Each such translocation event moves the mRNA through the ribosome by exactly one codon length.
This slide shows a diagram for initiation. We see two tRNAs. The first tRNA, labeled 1-tRNA, has the anticodon UAC and is carrying an amino acid AA1. It is bound to the start codon AUG on the mRNA. The second tRNA, 2-tRNA, with anticodon GAD is coming in. The diagram uses hydrogen bonds to show the base pairing between the codon on mRNA and the anticodon on tRNA. This visualizes how the correct tRNA is matched to the mRNA code during the initiation step. Next, a diagram for elongation and peptide bond formation. Now we see three tRNAs involved. The first two are bound and a third tRNA, 3-tRNA, is arriving. A peptide bond is being formed between AA1 and AA2. This shows the moment the chain grows. The anticodons of the tRNAs are correctly base paired with their respective codons on the mRNA strand, ensuring the amino acids are added in the right order. Slide 14 lists the steps of translation elongation in a clear sequence. Step 1. The eEF1 complex brings the amino acyl tRNA to the ribosomal A site. Step 2. If the codon-anticodon fit is correct, it triggers GTP hydrolysis, locking the tRNA in place. If the fit is incorrect, the tRNA drifts away. This is a proofreading mechanism. Step 3. Peptidyl transfer occurs, catalyzed by rRNA in the large subunit. Step 4. eEF2 promotes translocation via GTP hydrolysis. Step 5. The ribosome moves precisely one codon downstream. Step 6. And the cycle starts again for the next amino acid. This is the efficient, cyclical process of chain elongation. We are now looking at another diagram showing elongation in progress. We see that a peptide bond has formed between AA1 and AA2. The first tRNA, 1-tRNA with UAC, is now leaving the ribosome because its job is done. The second tRNA, 2-tRNA with GAD, is in the P site, and the third tRNA, 3-tRNA with GAA, has just entered the A site, bringing in AA3. The mRNA sequence below shows the codons being read AUG, CUA, CUU. This shows the ribosome has moved forward. The diagram on this slide shows the chain growing further. Now we have 4 tRNAs. A fourth tRNA with anticodon GCU has entered, bringing AA4. The polypeptide chain now includes AA1, AA2, AA3 and AA4, all linked by peptide bonds. The mRNA sequence shows an additional codon ACU, which corresponds to the newly arrived tRNA. This illustrates how the ribosome continues to move and the chain keeps getting longer. This slide seems to be continuation of the diagram. It shows tRNA2 GAD leaving the ribosome, tRNA3 GAA and tRNA4 GCU remain engaged. This is part of the cyclical process. tRNAs enter the A site, move to the P site as the chain grows, and then exit from the E site after donating their amino acid. Here, the diagram shows the polypeptide chain has grown further. We see tRNA3, GAA, and a fifth tRNA, 5-tRNA with anticodon UGA, which is bringing in the next amino acid. The chain now includes several amino acids linked together. The mRNA codons continue to be read in sequence. This diagram is similar, showing tRNA 3, 4, and 5 bound with a polypeptide chain attached to tRNA 5. It emphasizes the continuous nature of elongation until a stop signal is reached. Now, this is an important slide. It shows the end of the process. We see a long primary structure of a protein with many amino acids from AA1 up to AA200. A tRNA is shown at a termination or stop codon on the mRNA. The message here is that elongation continues until the ribosome encounters a special codon on the mRNA that does not code for any amino acid. This is the stop signal. Now we begin explaining translation termination. It says termination requires specific protein factors called releasing factors, RFs in bacteria and eRFs in eukaryotes like us. The signals for termination are the same in all organisms. They are the termination codons in mRNA. And there are 3 of these stop codons, UAG, UAA and UGA. The next slide elaborates on termination. After many cycles of elongation, a nonsense or termination codon of mRNA appears in the A site of the ribosome. This codon is not recognized by any normal tRNA. Instead, it is recognized as a terminal signal by the eukaryotic releasing factors or eRFs. The eRF then causes the release of the newly synthesized protein from the ribosomal complex. So, the finished protein is set free. This slide lists the steps of termination in order. Step 1. The termination codon enters the A site. Step 2. There is no cognate tRNA for it. Step 3. The eRF3 and eRF1 complex enters the A site. Step 4. This complex stimulates the hydrolysis or breaking of the peptide bond that ties the finished chain to the last tRNA. Step 5. With no acceptor in the A site, the process halts. Step 6. Finally, the completed peptide is released from the ribosome. This slide shows us the end product, the protein. The end product of protein synthesis is the primary structure of a protein. This is simply a sequence of amino acids from the first to the last, here shown as AA1 to AA200, all bonded together by peptide bonds. This linear chain will later fold into a specific 3D shape to become a functional protein. Now the final slides compare mechanisms in prokaryotes and eukaryotes. Slide 25 talks about prokaryotes like bacteria. The first point, the initiating amino acid methionine needs to be formilated. That's why there are 2 tRNAs for methionine in prokaryotes. Second, ribosomes enter the mRNA at the AUG codon or near a special sequence called the Shine-Delgarno site. Third, no initiation factors are needed for the initial contact between ribosomes and mRNA in prokaryotes. Fourth, the small 30S ribosomal subunit can engage the mRNA before the initiated tRNA binds. These points highlight key simplifications in the bacterial translation system. The last teaching slide contrasts this with eukaryotes. First, here the initiating methionine is not formilated, there's only one tRNA for methionine. Second, ribosomes enter at the capped 5' end of mRNA and then move linearly scanning until they find the first AUG codon. Third, this process requires ATP and many protein initiation factors. Fourth, the small 40S subunit binds stably to the mRNA only after the initiated tRNA has bound to it. So, eukaryotic initiation is more complex and highly regulated compared to prokaryotes. Alright students, this covers the core process of translation from initiation to termination and a quick comparison between prokaryotes and eukaryotes. Thanks for watching.